Hello everybody, this is Dallin Thewison.
I'm Logan Wade.
This is Kent Charrington.
And we're here to present Lesson 3 group problems. So if we look at this first problem, you can see right here as well as right here. We're talking about the Engelmann syndrome. And this is an epigenetic inheritance. And so one of the male, because it says paternal right here, whenever you have a recessive genotype with dominant, the dominant is silenced. And so with the outcome that we receive, we get homozygous dominant, non-disease. One of the heterozygous will be non-disease. However, because we are referring to this silenced genotype, one of them will be a diseased, and then just the homozygous recessive will be diseased.
Okay, for letter B, this question is going to be in regards to part A, and so you've got a following cross between a heterozygous male and a homozygous recessive female, and so it's asking what the genotypes and the phenotypes of the progeny are. And so we've got the cross on the left side there, and when you do the cross, you get that 100% are going to be diseased, and that's because of the silenced for the male.
Okay. And then on to letter C, it says, suppose a woman has triple X syndrome, meaning she has 3 chromosomes. However, the XICS has been deleted from 2 of the X chromosomes in each of her cells. How many bar bodies are found in each of the woman's cells? So as we understand from the readings, bar bodies develop from an inactive X chromosome. And if you have 2 or more XICSs, that would mean that you would inactivate one of them. But because she has 3, all of them would remain active because there's only 1 XICS. So then you would have 0 bar bodies because all of them are active.
And then for letter D, it says, suppose you isolate a cell from a female cat with 2 X chromosomes. However, the T6 genes on each X chromosome are non-functional, while the cis genes on each X chromosome are functional. How many bar bodies are expected to be found in this cell? And just from the readings, we learned that the cis genes, as well as the T6 genes, they are complementary to each other, and so we would end up with 2 bar bodies. And this leads us to our next question.
All right, so going into fill in the blanks here. So for letter A, the SRY is an example of a holoandric gene.
Okay, letter B, we got MIC2 is an example of a gene found in both the X and Y chromosomes.
For letter C, it says the cross is made between insulin-linked growth male mouse and an insulin-linked growth negative mouse, or female mouse. What are the phenotypes? And we can get that as a dwarf as well as normal.
Okay, for letter D, in the sex-influenced pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other.
Letter E, we got genetic imprinting is an epigenetic process that occurs during gamete formation, while dosage compensation is an epigenetic process that occurs during embryogenesis.
F is one of the topics discussed in Lesson 2 involved the formation of double-stranded RNA molecules. What 2 molecules form the 2 halves of the double-stranded RNA? Well, this is a cis RNA as well as a T6 RNA, and this is because one of the examples is what we did in problem 1.
Okay, so for the final one, we've got when reciprocal crosses give identical results, the alleles that control the trait in question are located on autosomal chromosomes. However, when reciprocal crosses give different results, then we know that the alleles are on the X chromosome.
Okay, so then leads us to question 3. Okay, so this is talking about a woman's father who has the OTD syndrome, I guess, I don't know, X-linked recessive disorder producing mental deterioration if not properly treated. The woman's mother is homozygous and is unaffected, so that would mean that she's homozygous dominant because it's an X-linked recessive disorder. Okay, so that would mean that the father's genotype, as you can see here in the notes, his genotype is going to be a Y with an X and a recessive linked X, okay, while the mother's, like we just said, is a homozygous dominant. So that's what we got for letter A.
For B, it asks if the woman has children with a man who does not have an OTD, what is the chance for the first child will be the son with OTD? And so with the first, or with the mother, we know that she's heterozygous, as well as the father. He is, the X-link on his is dominant, and so it doesn't have an effect on him. And so if we were to do this cross right here, we notice that because the female is heterozygous and that the X chromosome is effective, that would be passed to 25% of her sons in that cross.
Okay, for C, it asks us to identify the genotype of a man with whom the woman could produce a daughter with this condition. And if you kind of look back at number B, you notice that if the male does not have the condition, then the daughters, they can't have it, they're only carriers. However, if he does have the condition, then it's possible to give it to her daughter because she is a carrier.
Okay, so then in letter D, it says, consider the cross described in part C of this question. What are the expected genotypes and phenotypes of the offspring? So we just run a cross there, as you can see here. We got that 2 of them will have the condition, and then the other 2 will only be carriers. And so that's, as you can see in the cross there, that's what we would get. And then, yeah, that's deep. Yeah, that's it, right?
Okay, so with question number 4, we're so inexperienced at this. With question number 4, we're asked, the presence of a beard in some species goats is determined by the autosomal gene that is dominant in males and recessive in females. And so part A asks, suppose that a cross produces a 3 beard with 1 no beard phenotype ratio among male offsprings. So what we're looking for on this one actually is we have to be dominant, either homozygous dominant or heterozygous dominant. And so if we were to do a cross with a heterozygous male and a heterozygous female, we would end up with the results of 3 beard and 1 non-beard.
Okay, so for B, it asks us that the cross produces a 1 to 3 beard to no beard in the female offspring. And so it's going to ask for what the genotype and the phenotype of the parents are. And we actually got that they're the same. And that's because the beard and the no beard is actually. Let's see.
Okay. Heterozygous.
Yeah. Is that homozygous?
Heterozygous.
Heterozygous.
Yeah, heterozygous. So both parents are basically going to be the same because it's reversed in the sexes.
Yep. Okay, so as we go on to part C there, it says, suppose a cross between a male with a beard and a female without a beard produces a 1-to-1 ratio. Among the female's offspring, what are the genotypes of the parents? Okay, so among the female offspring would mean that if it's a 1-to-1 for the females, one would have to be homozygous dominant and the other one would have to be either heterozygous or homozygous recessive. So we determined that the father would have to be homozygous dominant and the mother would have to be heterozygous in order for us to get the appropriate ratio. Where's the... Oh, right there. Yeah, as you can see in the diagram there.
With part D, it says, suppose a male and female both, which lack beards, produce 1 beard, 1 beard and 1 no beard ratio among the males and females. What are the genotypes? And so if we were, amongst the males, what are the genotypes of the parents? And so if we were to look at this, we need to have 50% with beards and no beards, and so we need to have a homozygous recessive and at least a heterozygous. And so we can conclude that we need a homozygous recessive male, non-beard, as well as a heterozygous non-beard female in order to make this cross.
Okay, so that takes us to question 5, and we've got kind of this long paragraph here about cats, and it basically asks us, if suppose you cross an orange male cat to a tortoiseshell female cat, what are the genotypes and phenotypes of the progeny? And so with the diagram, you set that up. Of course, the male only has 1 X, which is going to be the little b, with the Y, and so you're going to get the males are going to be 50% black, 50% orange. I wonder if they can hear the piano.
They what?
That piano's probably pretty loud.
Oh, yeah.
Okay, anyways, the females, 50% of the females will be tortoise shell, black and orange, and 50% will be orange.
Okay, so then part B. So this concerns the trait described in part A. Male tortoiseshell cats are very rare. What is the genotype of a male tortoiseshell cat? So in order for a male tortoiseshell cat to happen, the male would have to have both the dominant and the recessive allele, which is hard to do because males only have one X chromosome. So the only way we could think that this would happen is if the male presented with an extra X chromosome, so it would be a XX, and he had a heterozygous mix of the dominant and recessive alleles. That way he would have the tortoiseshell phenotype.
Okay. In part C, we're dealing with the fruit fly Drosophila. A mutation produces a scallop phenotype, irregular margins on the wings. Assume that the scallop phenotype is the result of an X-linked recessive mutation. Indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. So it's important to note that reciprocal crosses is you're using a homozygous dominant in this case, since it is an X-linked recessive. So we have, as we see here, we have a homozygous dominant, and we make a cross with one that is affected, and then we get the results out. And so we'll get the results is we'll receive a homozygous dominant result as well as a heterozygous for normal girls, and then for males we'll receive just a dominant for them. So 100% will be normal and whatnot. So information.
Pause for some interlude
Yep,
music.
and then in the reciprocal cross of that, you're going to have 100% of the males affected and 100% of the females will be carriers. So you know that that's going to be X-linked because the ratios are not the same. Is that what you said?
Yeah.
Oh, should I do D then?
Oh yeah, so D. Yeah.
My man, follow along. Okay, so then, all right. So then on D, we're doing the same thing, but they want us to assume that it's an autosomal recessive mutation now. And so as we kind of talked about, with X-linked recessive, you're gonna have different genotypes and phenotypes with the reciprocal crosses, but with the autosomal recessive ones, we should have the same outcomes on whichever reciprocal outcome we do. And so we kind of just took a father who was homozygous dominant and a mother who was homozygous recessive who was affected, and we did that cross, and then you would switch it, which is essentially just the same thing, just different spots. And so either way, we got the same outcome.
Thank you so much. Have a wonderful day.
Hope you enjoyed the music.